picture this. You decide to start eating a high-fat diet.
Right. Okay.
And, I mean, you expect some obvious changes, right? A shift in your waistline, maybe an uptick in your blood pressure, or, some general sluggishness.
Yeah, the stuff we all kind of anticipate.
Exactly. But what you absolutely do not expect is that right this very second, that dietary choice is actively deleting the microscopic sensory nerves in your face.
Wow. Yeah, it sounds completely bizarre.
It sounds like a medical mix-up, like, I don't know, two completely different patient files got dropped on the floor and stapled together by mistake.
But it's really not a mix-up at all.
Right. It is not a mix-up. Obesity is quietly rewiring your facial hardware.
And this brings us to the source we're looking at today.
Yeah.
We were breaking down this May 2026 article in Science Daily.
Yeah, which covers some pretty wild research.
Oh, absolutely. It covers a monumental study published in the journal Nature by researchers at Helmholtz Munich and the Ludwig Maximilians University of Munich. And, I mean, the sheer scale of what they uncovered here, it fundamentally shatters how we usually think about weight and diet and just human anatomy in general.
So the mission for our deep dive today into this source material is to figure out how scientists even managed to, spot this invisible connection.
Because it wasn't easy.
No, not at all. To find it, they had to build this revolutionary AI tool that mapped an entire mammalian body, literally down to the cellular level.
Which is just incredible to think about.
So we are going to explore how they pulled off this, honestly, sci-fi level of mapping and, what they uncovered about the hidden systemic damage triggered by body fat.
And importantly, how this could permanently change the whole trajectory of human medicine.
Right, because, traditional medical research kind of operates with this severe blind spot, doesn't it?
It really does. It's like the old streetlight analogy.
Oh, right, where you only look for your lost keys where the light is shining.
Exactly. If you want to understand, say, a massive citywide traffic jam, but you only have a camera pointed at one single intersection, you are never going to solve the problem.
You're just looking at a few cars.
Right. And in medicine, if you are studying a liver disease, you typically slice up a liver and put it under a microscope.
And you just ignore everything else.
Pretty much.
Yeah.
You remain completely blind to the biological traffic jams happening simultaneously in, the nervous system or the immune network or the skin.
So this research team basically decided to stop looking at single intersections, and instead they put a satellite in orbit to view the entire city at once.
That's a perfect way to put it. They turned on the floodlights for the entire neighborhood.
Which is fascinating, but if you're listening to this, you might be wondering, how do you actually get a God's eye view of a biological organism?
Because you can't exactly ask a living mouse to hold perfectly still in a high-resolution MRI machine for 3 days.
Right, you're not getting cellular-level details like that. So to understand this discovery about the facial nerves, we have to unpack the mind-bending technology that got us there first.
And the main challenge there comes down to basic physics, honestly.
How so?
Well, to see individual microscopic cells across an entire body, you need light to pass completely through the tissue.
Which bodies don't usually do.
Exactly. Biological tissue is naturally opaque because it's just packed with lipids, so fats and water.
Right.
And when light hits those different substances, it scatters, which is why we are not transparent.
Right. So to solve this, the researchers developed this process called tissue clearing.
Yes.
And I read about this in the source, and my brain, short-circuited a little bit. They literally make the mice transparent.
It sounds made up, but yeah.
If I were standing in the lab looking at this, what am I actually seeing? A ghost mouse?
A ghost mouse is a very apt description, actually. So they use these highly specialized chemical baths over a period of days or even weeks.
Oh, wow. So it takes time.
It does.
Mm.
And these chemicals meticulously dissolve and wash away all the light-scattering lipids.
The fats.
Right. And simultaneously, they replace the water in the body with this synthetic fluid that has the exact same refractive index as the remaining protein structures.
Okay, wait. Refractive index? That just means how light bends, right?
Exactly. It's like taking a piece of heavily frosted glass and pouring a clear resin over it until it becomes perfectly see-through.
That is wild. So the underlying physical architecture of the animal is still there, but it just looks like clear gelatin.
Pretty much.
Yeah.
But before they initiate that clearing process, they tag specific targets in the living animal.
Okay.
In this case, they tag nerves and immune cells, and they use fluorescent markers.
So they make them glow.
Yes. Under standard light, the cleared mouse just looks like a faint gelatinous outline.
Almost invisible.
Right. But the moment you place it under the right lasers in a dark room, its entire nervous system and immune network just ignite.
Whoa.
Yeah, they glow vividly in three-dimensional space.
Okay, so we have this transparent, glowing ghost mouse, but I mean, even if it's clear, it is still a thick, three-dimensional object.
Absolutely.
If you want to map every single glowing cell, how do you look at the whole thing without slicing it into a million little physical pieces, like a deli slicer?
Right, because doing that would destroy the very map they're trying to preserve.
Exactly.
Yeah, slicing the tissue physically would completely ruin the spatial relationships between the cells.
So how did they do it?
Instead, they use a technology called advanced light sheet microscopy.
Okay, light sheet microscopy.
Think of how a traditional microscope works. It either shines a bulb up through a super thin glass slide, or it bounces light off the top of a sample.
Right, the stuff we used in high school biology.
Exactly. Neither of those works for an intact, thick object like a whole mouse.
Because it's too thick.
Right. So light sheet microscopy solves this by generating a razor-thin microscopic plane of laser light.
Oh, I see. Almost like a scanner beam sweeping across a document.
Yeah, exactly. But sweeping vertically through the three-dimensional space, it basically optically slices the tissue without actually cutting anything.
That is so clever.
The laser sheet illuminates exactly one microscopic layer of the transparent mouse at a time. And it completely ignores the tissue above and below it.
So you get a clean picture of just that layer.
Exactly. A camera captures that illuminated layer, then the laser moves down a fraction of a millimeter, and it snaps another image.
And it just keeps going.
Thousands of times. And then the computer algorithm takes all those individual optical slices and stitches them together.
Wow.
Yeah, it constructs a flawless three-dimensional digital replica of the entire organism.
But, I mean, if you are doing that at a cellular resolution for an entire mammalian body, the amount of data has to be absolutely crushing.
Oh, it's massive.
We are talking about tens of millions of individual glowing structures, right? How do they even process that without melting a supercomputer?
It's a huge hurdle.
Because, I mean, no human researcher could possibly look at millions of glowing dots and manually trace where an immune cluster ends and a nerve bundle begins.
And that data ocean is exactly where their AI framework, which they call MouseMapper, changes the game entirely.
MouseMapper, got it.
Yeah, creating the images was really only half the battle.
Yeah.
Interpreting them required computational power that frankly didn't exist until very recently.
Right. And when I was looking through this, I kept thinking of MouseMapper like Google Earth, but for biology.
That's a great analogy.
Because with Google Earth, you know, satellites stitch together the globe, and you can zoom all the way out to see entire continents.
Right.
And then you can just double-click your way down to see a single oak tree in a specific backyard.
Exactly. And MouseMapper lets these scientists view the entire systemic biological network of
That's
mouse,
right. Yeah.
and then zoom all the way in to see a single glowing macrophage sitting inside a very specific layer of tissue.
The co-first author of the study, Ying Chen, actually provided a crucial insight into how this operates in the article. Oh, right, about the foundation model.
Yeah. She noted, and this is a quote, MouseMapper is built on a foundation model, which means it generalizes far beyond the data it was originally trained on.
Which is a huge distinction.
It is. The distinction between a standard AI and a foundation model is essentially the difference between memorizing flashcards and actually learning to read.
Let's break that down a bit because, you know, the term foundation model gets thrown around a lot in tech circles right now.
Yeah, it's a huge buzzword.
So how does this AI actually learn the biology?
Well, if we look at older artificial intelligence, teaching it to recognize a kidney meant feeding it like 10,000 images of kidneys.
Right, you explicitly programmed it to find kidney-like shape.
Exactly. But a foundation model operates on the same underlying architecture as today's most powerful language models.
Oh, okay.
So just as a language model ingests millions of books to learn the grammatical rules of English, which allows it to write original sentences it has never seen before,
Right.
MouseMapper ingested massive, diverse data sets of whole-body imaging.
Ah, I see. So it learned the biological grammar.
Yes. Instead of predicting the next word in a sentence, it predicts the next biological structure in a tissue layer.
That is fascinating. So by understanding those fundamental principles of how tissues organize themselves, MouseMapper can autonomously identify and segment 31 different organs and tissue types.
And it can spot anomalies too, right?
Absolutely. It spots structural shifts, cellular clusters, even in anatomical regions it was never explicitly instructed to analyze.
Which is wild.
It provides this completely integrated whole body analysis system that spots the subtle beginnings of a disease anywhere in the organism.
And so once they had this ultimate God's eye view of a mammal,
Yeah,
they realized they could finally solve mysteries that traditional medicine just couldn't touch.
because they weren't just looking under the streetlight anymore.
Exactly. They didn't just want a fancy map. They wanted to watch a complex systemic disease in action.
Right.
And that is why they chose to point this technology at obesity.
A hugely systemic condition. Yep.
So they took a group of mice, fed them a high-fat diet until they developed metabolic problems, and then ran them through the tissue clearing and the MouseMapper AI.
And the results from that were just immediate and visually striking.
What did the AI actually see?
Well, the AI detected widespread chaotic alterations in immune cell organization across the entire body,
Just everywhere.
everywhere. It mapped severe inflammation simultaneously flaring up in the fat tissue, the muscle fibers, the liver, and the peripheral nervous system.
So the entire system was basically sounding an alarm.
Totally.
But the major shock, the thing that the traditional medical streetlight completely missed for decades, was severe damage to the trigeminal nerve.
Which is a huge discovery.
And I have to admit, I know the trigeminal nerve handles facial sensation, but the article kept mentioning the trigeminal ganglion.
Right, right.
That word always trips me up. Is a ganglion just like a fancy medical term for the root of the nerve?
Yeah, you can think of a ganglion as a sort of biological command center.
The trigeminal nerve itself is this massive cranial nerve.
Right.
It's responsible for transmitting touch, pain, and temperature from your face to your brain,
Right.
while also controlling motor functions, like chewing.
Got it.
So the trigeminal ganglion is the clustered mass of cell bodies that actually control those long nerve fibers extending out into the face, and it sits just outside the brain.
And MouseMapper revealed that in the obese mice, the sensory nerves branching out from this ganglion had been significantly degraded.
Yes, visibly degraded.
The microscopic nerve endings were quite literally being pruned away, just shrinking back.
Which is shocking.
It is. And here's where I have to push back a little because intuitively this sounds completely absurd.
It does seem like quite a leap.
How does belly fat end up destroying a highly specialized sensory nerve in the face? What is the actual physical mechanism that bridges eating a high-fat diet and waking up with degraded facial nerves?
So the research team used a technique called spatial proteomics to locate that exact physical bridge.
Spatial proteomics? Okay, what is that?
Well, proteins are the molecular machines carrying out every function inside our cells, right?
Right.
Proteomics is the study of those machines. And the spatial aspect means mapping exactly which proteins are active in a very specific microscopic location.
I see. So they zeroed in on the area.
Exactly. They focus this technique directly on the toxic local environment surrounding the trigeminal ganglion.
And what did the proteins reveal about the fat?
Well, the analysis fundamentally shifts how we need to view body fat.
How so?
We often think of adipose tissue body fat as a passive storage unit for extra calories, like a biological closet.
Yeah, you just put the extra stuff there and shut the door.
Right. But fat is actually a highly active endocrine organ.
Oh, really?
Yeah. When a body accumulates excessive amounts of fat, that tissue becomes mechanically and chemically stressed. The fat cells literally begin to suffocate.
Wait, suffocate.
Yeah, and in response to that, they release powerful inflammatory molecules right into the bloodstream.
So the fat essentially turns into a rogue gland, just pumping out chemical distress signals.
That is exactly what happens. Those signals travel through the circulatory system, triggering a chronic systemic inflammatory response.
Everywhere.
Everywhere. And when those inflammatory molecules arrive at the trigeminal ganglion in the head, they activate local immune cells, like macrophages.
Okay, and usually immune cells protect things, right?
Usually, yes. But here, instead of protecting the nerve, these activated immune cells create a toxic, hostile environment.
Oh, no.
They literally stop supporting the delicate nerve fibers and start breaking them down.
Wow.
So the systemic inflammation acts as the physical bridge, It carries the distress of the gut all the way up to the command center of the face.
That mechanism is staggering. I mean, it means a dietary choice is creating a chemical cascade that travels up to the head and literally redraws the physical wiring of your sensory system.
It's a complete rewiring.
And this wasn't just some, abstract structural anomaly sitting on a computer screen. The researchers actually proved that this pruning had real-world consequences for the mice.
Right, through behavioral tests.
Right, because physical structure always dictates biological function.
Always. And to measure this, scientists use these highly calibrated sensory tests. They often involve incredibly fine, hair-like filaments pressed really gently against the whiskers or the facial skin of the mouse.
Right, because, you can't just ask a mouse if its cheek feels numb.
You definitely cannot. So, a lean, healthy mouse will react instantly to the slightest touch of that filament. It's a vital survival reflex.
Of course.
But the obese mice were measurably less responsive, their reflexes were blunted, they required significantly more pressure before they even registered the physical contact.
So they were experiencing clinical neuropathy, like real measurable numbness and loss of nerve function, simply because of a high-fat diet.
Precisely.
Now, finding this phenomenon in a glowing mouse is a fascinating laboratory breakthrough. But anytime we talk about animal models on this show, the million-dollar question for anyone listening is always...
Does this happen to us too?
Exactly. Does this happen in my body? Are human nerves being crooned away by weight gain right now?
And the researchers anticipated that exact question, and they moved their focus straight to human pathology.
Okay, good.
They took the precise molecular signatures they discovered in the mice, the specific patterns of toxic inflammation and the protein changes responsible for the nerve remodeling.
Right.
And they analyzed trigeminal tissue samples from human patients with obesity.
And let me guess, the human tissue matched the mouse map.
Perfectly.
Seriously?
Yeah. The exact same molecular patterns linked to systemic inflammation and sensory nerve degradation were present in the human tissue.
That is wild.
The biological cascade that shrinks the facial nerves in a mouse operates identically in human beings.
Dr. Doris Kultnecker, who's the senior scientist on the study from the Institute for Diabetes and Cancer at Helmholtz Munich, she had a quote that perfectly synthesizes the magnitude of this breakthrough.
Oh, I think I remember this one.
Yeah, she stated, "We revealed previously unknown structural and molecular changes in the trigeminal ganglion and its facial branches, and the same molecular signature was conserved in human tissue. This kind of finding simply cannot emerge from studying one organ at a time."
And her point there highlights the fatal flaw in isolated research.
Yeah.
Because if a team of neurologists had only been staring at, say, human liver biopsies to study metabolic syndrome, they would never have thought to check the facial nerves.
Right, the streetlight was illuminating the wrong block entirely.
Exactly.
And recognizing that they have illuminated the entire neighborhood now, the researchers didn't hoard this technology or, patent the data behind closed doors.
Which is huge for the scientific community.
It is. They made these massive whole-body cellular data sets publicly available online for the global scientific community.
So now a researcher studying autoimmune diseases in Tokyo or, a team looking at muscular dystrophy in Boston, they can just log in and explore these systemic changes across any organ system they want.
That is such a powerful resource. But releasing that data is really merely the opening act for where this technology is heading.
Oh, absolutely.
The director of the institute, Professor Ali Erturk, laid out an ultimate vision for MouseMapper that borders on science fiction.
It really does. His goal is to build truly realistic digital twins of biological organisms.
Now, the concept of a digital twin usually belongs in aerospace engineering.
Right, not biology.
Yeah, like when an aviation company designs a new jet engine, they don't immediately forge it out of titanium. They build a flawless digital replica, a twin, and they use supercomputers to test how it handles extreme heat or, bird strikes.
And if the digital engine explodes on the screen, nobody gets hurt, and they just tweak the design.
Exactly. So how does this apply to biology?
Professor Erturk is applying that exact engineering philosophy to human health. By utilizing these incredibly dense, whole-body cellular atlases, the goal is to create digital twins of living organisms in both healthy and diseased states. Once you have a perfect computational replica, you can run what are known as in silico experiments.
In silico, meaning the experiment happens entirely in silicon, like on a computer processor rather than in vivo, inside a living animal in a cage.
Exactly, because think about the current model of drug discovery.
It's slow.
It's incredibly slow. It involves isolating a chemical target administering a drug to thousands of mice, waiting months, and just hoping the mice get better without fatal side effects.
Right.
It's slow, expensive, and it often fails completely when applied to humans later.
So with a digital twin, you skip all that?
Pretty much. With a digital twin powered by a foundation model, you could introduce a variable computationally. You could introduce a genetic mutation or a new pharmaceutical compound, and the AI would simulate exactly how that change cascades through the entire interconnected ecosystem of the body over time.
Wow. You could basically hit play and watch a disease develop and fast forward.
Yes.
Yeah.
You could trace a complex condition like type 2 diabetes or Alzheimer's or cancer back to the very first microscopic domino that falls.
That is mind-blowing.
And then you could simulate 10,000 different drug interventions overnight to find the one molecule that stops that first domino from tipping long before widespread symptoms ever appear.
So it promises to radically accelerate the discovery of cures while drastically reducing our reliance on physical animal testing.
Exactly.
So if you are listening to this right now, what is the ultimate takeaway? I mean, beyond the awe-inspiring reality of tissue clearing, chemical baths, and AI models that learn the grammar of biology.
Right, bring it back to the individual.
Yeah, this deep dive really serves as a mirror reflecting the reality of your own body. We're culturally conditioned to compartmentalize our health.
We really are. We tell ourselves, oh, my diet only impacts my waistline, or my stress only affects my sleep.
But this research obliterates that compartmentalized thinking. The visual and molecular evidence is undeniable.
That's all connected.
The state of your gut is in constant, real-time molecular communication with the nerves in your face, the immune cells in your joints, and the blood vessels in your brain.
You are not a collection of isolated, independent parts that can just be treated in a vacuum.
Right. You are an incredibly dynamic, singular ecosystem where every choice ripples through the whole.
And that demands a fundamental paradigm shift in how we approach daily wellness and, honestly, how the medical establishment treats chronic illness.
You cannot isolate a single variable in an environment where everything touches everything else.
Absolutely not.
So let's do a quick recap of the incredible ground we have covered today.
Good idea.
We started by exploring how scientists literally turned mice transparent to solve a physics problem, right? Using the razor-thin lasers of light sheet microscopy to map tens of millions of cells.
We explored how MouseMapper, that foundation model AI, acted like a biological Google Earth to interpret that entire ocean of data.
And then we uncovered the shocking reality that a high-fat diet transforms adipose tissue into a rogue gland, which triggers systemic inflammation that actively prunes away facial sensory nerves.
A process that is completely conserved in human biology, too.
Right. And we ended on the revolutionary horizon of in silico digital twins, which could allow us to simulate and cure diseases on a computer chip.
And, this journey leaves us with a really critical, lingering question to consider.
What's that?
Well, we just learned that a seemingly standard high-fat diet can quietly, progressively prune away the microscopic nerve branches in our faces without us ever consciously realizing it's happening.
Right. The physical symptoms might just manifest as a vague, creeping numbness or a slight loss of sensitivity that we just brush off as the normal wear and tear of aging.
Exactly. So if our daily mundane lifestyle choices can silently rewrite our sensory hardware in such an unexpected, far-flung location,
Yeah.
what other hidden biological domino effects are currently unfolding right now across the vast, uncharted maps of our own bodies?
Wow. That thought alone completely changes how I view a simple trip to the grocery store.
Right.
The medical streetlight is finally turning off, and the systemic floodlights are turning on, revealing an anatomy far more complex and intricately woven than we ever dared to imagine.
It's an exciting time for science.
It truly is. Well, thank you for joining us on this deep dive into the source material. Keep exploring, stay curious, and always keep questioning the hidden connections in the world around you.